Hi everyone, welcome to meeting 19 of the M2D2 series. We are excited to have Philipp Seidl with us today. Philipp will be talking about how to do low data reaction prediction using modern Hopfield networks. So a bit about Philipp before we start. He's a PhD candidate at the Institute of Machine Learning, GKU Links. He has a double major bachelor degree in information system and medical engineering, and a master degree in bioinformatics. His current research focuses on how to leverage machine learning methods, especially transformers, modern Hopfield network, and featured learning algorithm for drug discovery application. Thank you so much, Philipp, for accepting to present today. And it was very short notice our invitation, and you still make it through. Thank you so much, and looking forward to your presentation.
Thanks for the kind introduction. Let me right away share my screen and see. Please respond if you can see my screen. It should be working. Let's start it.
Yeah, looks good. Thanks.
Perfect. So I'm really excited to talk to you today about our work. It's really cool that you have me here. On my presentation style, so if you have really urgent questions or I didn't explain something very thoroughly or it's a bit unclear, please unmute yourself and just ask, because it's hard for me to follow. If someone is raising their hands, just unmute yourself and talk. If it's not an urgent question, please wait till the end to ask it. There we will have plenty of time to discuss matters. So today I will be talking about improving few and zero shot reaction template prediction using modern Hopfield networks. This was work we started off in late 2020. And yes, so what are we trying to do here? Let's give you a bit of motivation on the topic. So we are trying to synthesize or make a desired molecule. So a molecule might be so we have a molecule on the bottom left here, which is from a sponge. It's called kappa-cahin A. And this deep sea sponge is very seldom. It's very hard to extract this molecule here, but it's through wet lab experiments, they have found that this is might be a very good candidate for cancer, but I think it's breast cancer. And there have been numerous teams and years of development of synthesizing this compound, and it's very complex actually to synthesize molecules. But it's a crucial step in molecular drug discovery. If you can't make it, you can't test it. Another motivation is that the chemical space is vast. Like there are those numbers out there that that say it's 10 to the power of 60s as of now, and that's the druggable space. So not just the chemical space, but the possible drug space. And as of now, we're in the range of 10 to the power of 9, 10 to the power of 10, dependent on where you want to buy your molecules. But it's tiny compared to the huge space that is out there. And we really would like to have a method that can tell us if really fast if a certain compound can actually be made. And how do we go about this problem? Computer-assisted synthesis planning has been around since decades. We try to tackle this problem from the end. So we start off with our target compound and recursively decompose it until we have just molecules that we can actually buy or have in stock. So how this works, you see on the bottom left here, we have a target molecule, and there are now those two arrows. Those are possible synthesis pathways. The red molecules are molecules that are not in stock, that are in stock, and the black ones we again have to synthesize. So in this first path, there is one molecule that is not in stock, and we stop here because we think it's really hard to do. Another reason might be that we have toxic byproducts, or even another one is that we have very low yield. So you can see this is a rather complex decision that we have to make here. and that the chemist then actually has to make. So in the second pathway, this molecule number 2 is again not purchasable. So we again derive it and have 2 possible synthesis pathways. For both, there is again a molecule which we can't buy. But then for molecule number 3, we find a synthesis pathway where we can start with molecules that we actually have in stock. And this whole process that we have done right now is computer-aided synthesis planning. And again, we want to find a path that leads to molecules that are purchasable and there are a lot of other constraints. But at the core is always this single-step retrosynthesis model that tells us how the arrows, so how to come from one target to reactants. Here is another depiction of CASP. In CASP system, you usually have a huge data set. There are multiple data sets out there. I will talk about a few of them later. And what we do is we have this template library in there. I will also talk about later about this in detail, but a template sort of describes the transformation of a reaction. And this knowledge base that we derive from the data set has a curated reaction sequences. There are 2 models that can be, or actually multiple tasks that can arise, and you can see this on the bottom here. So we can have conditions that we are interested in, but also the yield. And there is the forward path and the backward. So given reactants, we can try to predict the products and the other way around. And again, as I have said earlier, what's really crucial is this single-step retrosynthesis, because it's essential in each node of the right graph. You can see that there is a branching, and we really want to traverse this tree. This is our end goal, and find a path with certain constraints. One can, if we take the analogy of playing chess, in chess you have a rather low branching factor. For retrosynthesis, you have a very high branching factor, but then again, you don't have that many steps, because in chess you have, I don't know, a lot of steps, but in retrosynthesis you wouldn't usually consider, I don't know, beyond 10 steps, depending on how important that molecule is for you. But you want it to be done within 10 steps. Otherwise, you probably wouldn't consider it as doable. Yes, so the first distinction that is really important is again between reaction prediction and single-step retrosynthesis. The first one, reaction prediction, here we are given the reactants and we want to predict the product. This is also sometimes called as forward prediction. In the retrosynthesis or single-step retrosynthesis, it's the other way around. We are given the product, and now we want to predict the reactants that lead to this product. There may be multiple reactants that lead to a product. This is the same for reaction prediction. So there have been a bunch of approaches now which tackle this single-step retrosynthesis approach. And again, this is the core of computer-aided synthesis planning. In the literature, it's very common to distinguish between template-free and template-based. As always, it's very hard to push certain approaches into categories. Nowadays, there is also these semi-template based or also half reactions. It's known. Let's say it's not that easy to categorize them and push them into different categories, but here this template-free and template-based is a rather established categorization. So among template-free approaches, and again, I will later explain what templates are in detail, but one of those template-free approaches is sequence to sequence. And you've had, I don't know who listened to the talk about from Philipp a few weeks ago. He presented his work on transformers. This was a sequence to sequence task. And today I will briefly touch upon the topic. So I will talk about the translation, But again, on template 3, there are also graph-to-graph based approaches. And in the template-based approaches, we have expert systems where we don't even have a learnable system, but just execute the templates based on some rules. Then there are classification approaches which predict the reaction template given some learned model. One of the seminal works is by Seidl et al. Fortunato and also Dai, which is the GNN. And nowadays there is also a generation approach. So we not only derive the templates, but we generate them. I won't talk about those approaches. Those are rather recent and are still preprints. But just to note that those templates can also be generated. And at last, we have the retrieval approach, which is our work. Here we don't try to classify them, but retrieve them from a database. Yes, let's give you a bit of, let's talk again about template 3 approaches as Philipp Schwaller's talk. So I think as of now, everyone has heard about those transformer models. They have taken the NLP domain by storm. They're really efficient. Their parallelizability really gives them an edge over, for example, recurrent models. And they perform really, really well in this translation task and other NLP tasks. So it's quite intuitive to also try to apply this in the chemical domain, because here we also have a translation task. So in this work by Schwaller, they represented the molecules. Here they had reactants and reagents, and they simply inputted this into an encoder-decoder transformer architecture, and their target was to decode the most likely product. This is now forward reaction prediction, but the same has been done for backward reaction prediction. But their first approach was in forward prediction. There was also one of the early works by Liu was using an LSTM, but I can give you later more updates on how this method performed back then. So again, this in the forward setting is way easier than the backward setting, but this transformer achieved a really high accuracy of 90%, predicting the correct product. But a bit of a downside is that it's difficult to inspect. So there are no transformation rules. And to achieve really good performance, they need a lot of augmentation. Here you can do Smiles augmentation, but also switching the order of reactants, for example, is another augmentation strategy. Or yeah, there are multiple augmentation strategies here. And yeah, in order to achieve really good performance, those are necessary actually. This does not speak of good inductive bias. This is just an opinion, but if you have to do a lot of augmentation, like augmentation should not be necessary if you have good inductive bias of the model. Nevertheless, those models have really, really good performance. And what's really interesting is that they can be used for our approach too. So, but I will talk later more about that. So what are those template-based approaches? Or first of all, what's a template? So you're given a reaction equation. This can be from some database. And here I've noted the Smiles representation, but it's really ugly to look at for humans. So we really prefer this representation of molecules. And the first step that we do actually is we perform atom mapping. And really surprisingly, the transformers, although never trained, actually perform really, really good on this atom mapping task. So colleagues of ours have been working on an independent benchmark, and there the transformer architecture produced the best atom mapping. So after you have this atom mapping, which tells you the correspondence of the reactant atom to the product's atom, this might seem trivial, but it's quite a hard process to do this right. It's not always so clear cut that it is as it is in this example, but here you can see one chlorine, and you can see the correspondence between two. And you can also see immediately that along this nitrogen-oxygen bond here, there something changes. You can safely ignore the solvents here because they actually don't change anything in the reaction. So this is not captured in the template. But what is captured is how the atoms change as well as how the bond changes. So from this example at the top, we would extract a template around the reaction center. And there is also a hyperparameter, which is how far of the reaction center do we want to go? What's the radius around the reaction center? And usually people take a radius of 1, so around 1 reaction center. If you extend the radius, you get much more precise templates, but they are not so general anymore. So there is a trade-off here still. But again, those reaction templates can be generated from reaction equations found. But then again, there is also another way, which is a chemist sits down and thinks of those templates themselves. And yeah. Now, what's the task of reaction template prediction? You're now given this product here, and your task is to predict the index of this template. So which template to actually execute or apply. This might seem like a trivial task, but it becomes tricky because there are thousands of those templates, and for most of them, you have very, very few samples. So if we look here, those are two data sets, USPTO small and USPTO large. What you see on the bottom is the template popularity. So how many samples we have for this template. And you can see for a USPTO large, for 42% of the samples, we actually just have one sample for the classes, for the corresponding templates. So most of them are in this few-shot setting regime. And I will later talk about approaches on how to handle this and what has been done in the literature. After you have selected a template, the next step is to execute it. And again, with this example here, we are given the product. We take the reaction template, we run it. In this case, it matches the nitrogen and the carbon here, and it transforms it to this nitrogen oxygen. Next thing we have to do is check if it was the thing that we started with. And if we neglect the byproducts or the solvents, you can see after you look carefully, because it's switched, this is actually the same molecule.
Can I ask one question then?
Sure, anytime.
You mentioned that you check what you start with. So I'm guessing these templates are not foolproof, like they can actually...
Not at all.
Okay.
I will later talk about this more. So they are not at all bulletproof. They can match at different regions. Another thing is that, yeah, one template might produce multiple results, and yeah.
Okay, cool, thanks.
But in this setting here, we are trying to check if what has been produced by the template actually matches the one that we have in our database. Now, again, this seems like a very, very easy task. Why do we actually need machine learning for that? So one thing is that this application is rather expensive. Also checking the applicability. So applicability, what is that? Here you're checking, for example, here, this template here, we're checking if our molecule has a carbon and a nitrogen in there somewhere. And this is an NP-hard problem. It's quite computationally expensive if you do it on a very large molecule, for example, because there might be actually a lot of ways this could match. And actually, when you look at this, so at all your templates, a lot of them apply usually. And even though you can apply those templates, they might not be relevant. So what is relevance? Relevance actually tells us if we have found this template in our database. So we deem a template as relevant if the template, first of all, produces the results seen in the literature or in the dataset, and applicability is just a necessary condition. So I hope that that clarifies the huge distinction between applicability and relevance. And again, why is this a hard problem? Because there are so many templates that would actually apply, and the relevant ones, for those we have just very, very few data. And how has this been traditionally been approached? So the first approaches, for example, were by Y. There they only had 16 expert coded templates, so not that many. And they were the first ones to use a neural network for this task, also due to computational reasons. And then Seidl and Waller, in their Nature publication, they first started with automatically extracted templates. There they had 9,000 templates, but again, removed those where they only had less than 3 examples to train with. Later, Balon used the hierarchical grouping of those templates and trained separate neural networks for different groups of templates. Then in 2020, Fortunato pre-trained on this applicability that we were talking before. Training on the applicability already gives you a bit of information. So on the bottom, you see how this is typically handled. So you have a molecule encoder, you have some representation of the molecule, and here M of H is a hidden representation. And now the last layer, W here, is the weight matrix of the last layer. If you think about how this is trained, you get updates for this W matrix. Let's say the rows correspond to then the corresponding template classes. But if you don't have any examples, this will stay at the initialization. So it will stay at random. And this is not very well. So training this on applicability already gives you sort of gradient update on this W matrix, and you can learn something from the W matrix in the corresponding rows, even if you have few data changes. And then one other approach was by Hyde in 2021. There they grouped templates, and they had an algorithm where you can drastically decrease the number of templates by making them much more general. So how do we go about this problem? We are proposing a Modern Hopfield network for this. So in contrast to other works where they used this random W matrix, we actually use the structure of the reaction templates and encode it as well. There are multiple ways this whole approach can be seen. We view it as a Hopfield networks, but one could also view it as, or it has a lot of similarities with contrastive learning approaches. Yeah. So again, a lot of you might not be very familiar with Modern Hopfield networks. But Hopfield networks may ring a bell. So Hopfield networks have been around since the, I think, 70s or 80s. So they're quite old. They have been used as a model for memory, and they're also used for modeling quantum states. So the spin model, the Ising model is a Hopfield network. And if you change back then this was binary, but it has been since then extended to work also for continuous cases. And if you use this continuous case, and also if you derive this new energy function for the Modern Hopfield network, and the update rule that corresponds to it, if you look at this formula that is derived from this new energy, then this is surprisingly similar to the attention in transformers, which is really striking. And we were working on this with the paper Modern Hopfield Networks is All You Need. And we were trying to get insights so theoretically insights, and use them for training transformers. So how can you use those Modern Hopfield Networks? Yes, sure.
What will you say is the biggest advantage or disadvantage of Modern Hopfield network compared to transformers?
Yes, so the you can't really say disadvantage or advantage, because if you look at the formulas, they're similar, very similar. So the only difference is that there is a relationship between K and V, and actually there have been papers which model K and V, so the key projection and the value projection, they have a shared key and value projection. So not even so before we proposed our modern Hopfield networks. So you can't really say advantage or disadvantage. This is just another way of viewing the whole problem, and we hope to gain insights from this theoretically, because modern Hopfield networks or Hopfield networks in general, they have been around for such a long time. And the main argument is rather that we hope to gain insights from the theoretical views of Hopfield networks. I hope that clarifies the question.
Yeah, it clarifies it. Thank you.
Thanks. So let me show you one example of how to use those modern Hopfield networks. So here we have a really illustrative example taken from a blog. And what we start with is this R here, which is a noisy version of Homer. And we have a database. So these are our stored patterns. And here the noisy version of Homer is the state pattern in Hopfield lingo. And we're trying to retrieve Homer again, even though it has been very noisy artifact. So what you typically do, and this is not represented here, but you would map it down to some association space. So you have a projection. This is not shown here, but you can think of it like this. And we're computing the dot product. And out of this softmax of beta times R y transposed, what we get is a probability over our database. And what we then do is we take this probability and multiply it again by our database. And if this probability is like 1 at 1%, so if we have chosen the right beta here, because beta is really, really crucial in this setting here. If beta is rather high, then we are very concrete, and we only take one sample out of this database. In this case, we can recover the phase of Homer again. So to take this back to our setting, we have reaction templates, and the reaction templates are database from which we want to retrieve, and the state pattern, so this noisy image of Homer here, this is actually the molecule that we input and where we want to know which template to use. And what ends up happening is that we learn an association space. So the association space is the space in here where we have X transpose Xi. So we learn a space where the similarity between molecules and templates is such that we select the templates which are most applicable, most relevant, sorry. Yes. Is that clear? Should I clarify this more? Or otherwise I will switch over to our experiments section. So we've done extensive experiments, mostly with the USPTO dataset. I will talk later a bit about the USPTO dataset. There are two, yeah, there are raised hands. Thanks, please speak right away.
Yes, can you go back to the previous slide, please?
Which one? This
the one with the reconstruction of the picture.
Yeah.
So basically you consider this as more like a retrieval problem, not a generative problem.
Right.
Okay.
But you could equally, like if you've generated new templates, you could add them to your database, which is a very good comment. But this is seen as a pure retrieval. So we have our fixed database. Our database is the templates that we extract from our samples. And yes, and we try to retrieve it from that. And I will talk about a bit more about how we designed the experiments. So is there another question, please?
Yes.
Yeah.
Okay, go ahead.
Oh, for me? Yeah, can you go back to this slide? So you said when the beta is big, there will be a concrete just one output. So what if the beta is small? Will it be
then you will get a mashup of all the Simpsons characters, and this will be the pattern you receive. will be a mixture of the different pictures in your database. So we can see like this beta parameter is like the inverse of the temperature. If you think about temperature parameter in Softex.
Okay, thank you.
And this is, I want to emphasize here again, this is just one application how modern Hopfield networks can be used, and we have used it here.
So one question, go back to the previous slide again.
Yeah.
Thank you. So it's probably a comment, but yeah. If you are using encoding the molecule and then encoding the template and trying to match the molecule to the template, I think what you're actually doing is restoring the representation of the molecule to the part that is active or kind of participating to the reaction, right? And the question then is, what happens if there's different reaction possible, like template possible? How does doing this kind of affect negatively the whole process?
That's a very, very good question. So what happens if, for example, there are multiple templates which would be equally viable, or so this should actually be captured in the, so this is one of the core assumptions here, that there is some structure within the reaction templates that corresponds to the molecules. So if we have a certain input relationship, then this will translate back to a certain, I mean, dependent on how nonlinear you make this here, but it will always translate back to the probabilities you will get when inputting a molecule. You sort of have this, again, if you think about the W matrix here, like here in this approach here, like others have done, the W matrix does not restrict your problem at all. You can change the W matrix however you want. But in our case, dependent on the representation you have here for the reaction templates, this then is corresponding to your probability, or this corresponds to your ranking. If you have two very, very similar templates in this input space, they will be also very similar in the prediction output, in the ranking. Has that answered your question? If not really, right? You asked also if that negatively, if that impairs actually the performance, if you have.
So let's say this way: if you have one molecule that can be used by two templates that are very, very different, I think that's when the problem arises, right?
So you have one molecule and actually two templates that would be equally well, right?
Yeah, but they are very different in terms of structure.
Yes, then over time, the learning occurs. It should learn that the association vector is in between those two. The thing is now, we assume that the template vector, so the association space, like if we assume this is just a ball in the 2D case, if they would be very structurally dissimilar, this would actually be a disadvantage, because what the model would learn over time is that molecules gets mapped in between those two. And then it might actually impair it, but we have not, one might design, because this association space is not just two-dimensional, it's in our case 512 or 1024, you might find a dimension in which you can put this molecule such that it improves both. So it's within a space where you have where the molecule gets mapped, where it corresponds to both of the templates. If you have for the templates the same, if you have both of them in your training data set. If not, you can't learn this relationship.
Okay, cool, thank you.
Okay, so how did we design the experiment? There is a huge distinction between template relevance prediction. Here, I will talk about later about this, and also, sorry. So we first of all took the USPTO dataset, because USPTO dataset is publicly available, and this makes our results reproducible. We also shared code, so people should be able to reproduce our results. For the template relevance prediction, which is one of the tasks, we used pre-processing and split based on Fortunato, because this is the approach we compare against in this setting. For both, we train at the maximum of 100 epochs, and we used Bayesian hyperparameter tuning. I think it was Optuna that we tried out, but manual hyperparameter tuning turned out to be better in our case. Of course, we selected the best model based on the validation performance, and in our case, it was the top one validation accuracy. How did we extract the features? So in our case, we used fingerprints, but not just Morgan fingerprints. We used multiple types of fingerprints. We also used the RD kit, also a newer variant, which is MHFP. So we used multiple fingerprints. One could equally try this out with a GNN. For the templates, we also used fingerprints, but here it becomes more tricky because you have a variable size of reactants, but one product in the template side. So here we have calculated the fingerprint or the structure of the template. And we found this by trying out different combinations. So you take the fingerprint of the product side and you subtract a pooled version. In this case, it was max pooling, or it's a hyperparameter, which pooling operation you choose, but you pool over the fingerprints of the reactants. And what's also important is that you add noise if they are frequent, or add, like, the noise allows you to have some learnable component for very frequent templates. And yeah.
How did you, just a short clarification question, but how did you use the multiple fingerprints for the molecules? Were they concatenated, or did you consider them a hyperparameter and you picked one of them?
No, the multiple fingerprints are just added. So okay. The cool thing about the fingerprint is that the information is spread around in this vector. And even if you add, the information still preserves under a certain condition. Like, okay. Because they're randomly, you can just add them and still obtain more information. I mean, it always will depend on how big you allow your fingerprint to be. But in our case, I think it was 8,000 something. So the fingerprint vector is quite large.
Okay, check.
But again, it would be interesting to try this out with a GNN. Actually, one of my students is working on this, how that improves, or how that changes performance, because you might, so for this task, it's really important to get the details of the molecule, and yeah, might be better captured with a GNN. But empirically, this worked very well with using these different fingerprint steps. So we were using this USPTO database dataset. It was parsed from patents and was done in 2012 by Loeb, and contains roughly around 2 million reaction equations. The tasks that arise are actually quite, so it's really cool data set. You can just do a single-step retrosynthesis, but also forward synthesis is done on this. A very challenging task is the yield prediction, also because the yield varies much by who is actually performing this reaction, but this nevertheless is a task that is performed. Another task that may arise from this data set is template relevance prediction. Again, very similar to single-step retrosynthesis, and reaction class prediction. So chemists categorize different reactions to different classes, and this can also be one of the tasks. But also a very challenging task would be to predict the conditions, which we are currently, within our approach, we are not modeling. We just model that this reaction would occur under some conditions. Yeah, this is actually a very important remark here. Currently, most approaches don't model any conditions. So these reactions, they only happen under certain temperatures. There has to be a certain process involved. As of now, this is not modeled. Also, it tells us that this transformation is somehow possible. So we know that there are some conditions under which this reaction might undergo. And what it also does, it leaves out possible intermediate states. So we only can measure what happens at the end, and there might be some intermediate states which are not captured. And yeah, a few more remarks. So we have 4 subsets of this USPTO and 2 core tasks, which are template relevance prediction. In template relevance prediction, this is in the setting of also Fortunato et al. Used. We only use the samples where we from valid templates. So if we execute the templates, there should be, at least it should be possible that we can get the right product or the right reactants out of this product when applying the extract template. So in USPTO small, we are left with 40,000 samples. And for USPTO large, we have 440,000. And here you can also see the big difference in amount of templates. So there are 11,000 templates, and a template is unique because it might also not be straightforward. A template is unique if it has a unique string identity after canonicalizing it and also removing the atom mapping. So removing the numbers corresponding to each of the atoms. Yes. The metric that is used for template relevance prediction is if the template is among the top K ranked ones. This is very much different to the metric that is used to single-step retrosynthesis. Here, we not just look if the predicted template is among the top K ranked ones. Here, we want to actually apply the templates that we predict, and the results of this after we have applied them. This now counts. And as I've told you that if you apply a template, there might be multiple outcomes. Yeah, this gets counted here. So yeah, and this now contains. Sorry, yeah.
Sorry. So the first template relevance predictions, is it a forward synthesis?
No, they're both backward synthesis. Sorry, that didn't clarify that. They're both backward. So single-step retrosynthesis in their task, right? That's a good question. Template relevance is also retrosynthesis, but just on a separate set where we know that the templates actually are applicable. The single-step retrosynthesis is a way harder task because here you actually want to know if you apply the templates, do they actually produce the right result? Yeah. And here we have 2 subsets, USPTO 50K and full, again, the larger. If you remember, USPTO has around 2 million. A lot of them are error, or duplicates are also a few of them. So we used the pre-processing by TEDCO, which is left with 1 million samples. And if you extract the templates, we arrive at around 200,000 templates. And again, what's also important here, we only use the templates in this setting from training and validation set. This is also different from the template relevance prediction. Okay, so previous work, again, as I noted, Fortunato et al., where you have this W matrix at the end, this would correspond here to these connections at the last layer. And here, because they are trained on the applicability, they get some sort of information. And what's really cool is you can do zero shot with them. So if you look here, when you pre-train on applicability on the examples where you have 0 examples, so this is a zero shot setting here, you get a top 100 accuracy of around a little bit below 10%, which is really cool. So it's way better than the baseline, which is a neural network where the W weights for set zero shot examples would stay at the initialization. If we look at one shot, we also get quite an improvement over a baseline. And this is not true if we look at classes or templates where we have more than 50 examples. So here on the bottom right. But still, this is these results can be improved. And if we look at our approach here by using the structural information, so our approach is the one in red here. And by using this structural information and thinking of this problem as a retrieval problem, we achieve a 80% top 100 accuracy in the zero shot setting. And again, you can see here with the gray bars in the background that actually the biggest amount, also the most amount of samples in the data set are actually in the setting between 0 and 1 shot. So yeah, This is really, really cool. What's also interesting is we have looked at the popularity filter. So here you would just execute the templates based on how frequent they appear. And then you add a fingerprint filter, which also was where we were the first to use this very cheap and computationally cheap approach of filtering out non-applicable templates by their fingerprint. So if the product side fingerprint does not match the fingerprint of the product of the input product, we can safely say that it is not applicable because the substructure is not in there. And through this very cheap filter and popularity, you are also very good at this setting where you have more than 49 samples. But of course, in this few shot setting, you're poor, you're very poor. But this is a very straightforward baseline that actually performs reasonable in this setting where you have a lot of templates. Yes. So let's get to the second task, which is way harder. This is single-step retrosynthesis, and this is a very crowded benchmark because here a lot of methods have been proposed. Again, here we see on the top the methods that we have implemented, and you can see the category. They're categorized into TB, template-based, and template-free. And on the top right, we can see here the top 1 accuracy here corresponding to if the reactants that are obtained after execution of the template. And you can see here that in the top 3 regime, so if you look at top 3 accuracy or more than 3, we are state of the art here in this regime, and we achieve, for example, in top 5, achieve 81% top 5 accuracy, which is really astonishing. So when we started, we would not have thought that we get into this, get this high, because there are so many other methods which are doing really, really cool stuff. And for example, if we talk about dual TB, what they are doing is they have this energy-based model on top, and they are doing re-ranking on the outcomes that have been produced by templates in this template-based setting. So with this re-ranking model, so we're in the top 3 setting, we're as good as they are with their re-ranking method, which is really, really cool. Also, if we look here, ATX100, this is an augmented transformer variant, which has 100 times augmentation, and it performs also in the range of around 81% in this top 5 setting, which is also very good, but again, needs augmentation, needs a lot of augmentation. Yes. If we look at the USPT04, where we have many more templates, so here we have around a range of 260,000 templates. Those are extracted using the RD chiral package, but one could also use, for example, a transformer, and this might actually improve the results. So in this setting, we're on par with the AT. In some settings here in top 10, we're a bit below. I don't know why this is the case, but yeah, it's not that different. So here we're still very competitive here. But our main selling point, so this single-step retrosynthesis is a method that is actually applied a lot of times. And what's really cool is that we're orders of magnitude faster than other approaches. So on the bottom here, we have a log scale of seconds it takes per reaction. And if our criterion is the top 1 accuracy, so in our case, we just stop after the first submission of our molecule. So the first valid molecule that comes out of a template, and then we stop. And yeah, in this diagram, you always want to be on the top left part here. So you want to have a fast method, or you would always take a faster method that gives you a better accuracy. And you can see here that we're orders of magnitude faster, and yeah, while having higher accuracy. But I also want to give you some remarks on accuracy, and Philipp Schwaller also noted this in his talk. So top 1 accuracy is usually chosen here as the benchmark criterion. And if we think of ImageNet, this is actually standard nowadays. Back then it was not standard, so there were also top 5 or top 10 accuracy, what people looked at, but nowadays they only look at the top 1 accuracy. But in this setting, let's think about what actually happens if we have multiple valid answers. So let's assume that we have, for each of our products, we have 2 valid synthesis ways. In this case, the maximum top 1 accuracy can only be at around 50%. And this can also be shown empirically, that this top 1 accuracy does not always necessarily correlate with the multi-step planning evaluation, because for multi-step planning, you also need a lot of diverse and you need a lot of synthesis paths to explore. Yes, at the end, I want to also give you a remark on symbolic rules. So let's zoom out a bit, because reaction templates are essentially a symbolic rule, and this does not apply only in the chemical setting, but to so many other settings. So the reaction template actually is a recipe for the transformation. It describes what is happening. And it can be designed by an expert, as I already noted. It can be extracted from a database, but it can also be generated. And one of the problems that is always noted with those symbolic rules is that, those rules, they do not generalize well. So if we look empirically, we get very high performance, even with those simple symbolic rules. And as I noted also, it's very much dependent on the reaction radius. So you might want to have a very small reaction radius, but then again, those rules are not that specific anymore. And what's also a bit of a problem is that the rules might not be applied in the right way, as I noted. So if we have very unspecific templates, they might apply at multiple positions. But one of the core benefits of those symbolic rules is that they're interpretable. So chemists really like to stay within certain safe boundaries, because yes, a model might suggest that there is a path within which you could derive it, but if you can't tell the conditions, or it's a very rather strange path, the chemist might not look at it because it's too novel, and it's much more safer with known reactions. Also, what these reaction templates, or in general those symbolic rules, can represent is something very complex, if something very complex is underlying. And this is, for example, the case in metabolism. So metabolisms, you have a protein that facilitates, or is an enzyme that facilitates a certain reaction. And this reaction can be very, very complex, and it's very hard to model it otherwise. So the templates could, a template could model this. Yeah, they're indeed used, for example, in metabolism. Yes, it's just like blue, okay. At the end, let's conclude and summarize my talk a bit. There have been several approaches that I presented. The first ones were, for example, the expert systems. I would also call the popularity based with the fingerprint filter an expert system, because it's also not learned. Those are overly simplistic and there is no learning, but yeah, in certain settings they achieve quite good results. But yeah, the translation approach show very good predictive quality, but are not that interpretable, and might lead also to results that are not that straightforward, that might be out of the boundaries that the chemist is comfortable with. Then there are classification approaches. Here, the classification approaches are usually weak in the few-shot setting. But yeah, seeing this as a retrieval, we combine previous approaches. We have very good predictive qualities, especially for templates which are unseen or rare. We're computationally very fast, and this can be used for even with new templates, for example, that chemists would design without retraining the model. And yeah, code is online, and I urge you to play around with it. And yeah, that's it. Thank you all for listening. I want to thank the collaborators that were all working on this project, also the sponsors, and also the colleagues from my institute for the thorough discussions we had. And yeah, if I could see farther, it's because I was standing on the shoulders of giants. I want to also say thank you to the community, because I mean, nowadays it's so easy to plug in a model and train it, but yeah, back then it would not be possible without the awesome community that is behind it and builds tools that everyone can use. Yeah, thanks. And I hope there are a few questions. Otherwise, yeah.
Great. Thank you for the nice presentation, Philipp. I really enjoyed. Is there any questions?
Otherwise, I might take a few. Sorry, is there a question?
No, I don't see anyone.
Philipp?
Yeah.
Yeah, thanks for the presentation. I just have a
Thanks.
question for the augmentation you mentioned earlier. So what kind of augmentation do they normally do?
The augmentation for transformers?
Yeah, or for like, I think you also mentioned other pre-training with augmentation.
So the augmentation, I don't need a slide for this. So the augmentation for transformers is usually a smiles augmentation. Smile string is not unique, except if it's canonical smiles. But what you usually do is that you take the smiles, which is a linearization of this graph, this molecular graph, and there are multiple linearizations. And what you can do is train it with those multiple linearizations. So you would tell RDKit to not give you the canonicalized version of the smiles, but give you a random starting point from the graph, and you could do this, iterate over it, get multiple predictions in. You can do this in inference, but also in training. And it has been shown a lot of times that this helps a lot in transformers. And in this case, it's not only the position, not only the canonicalization, but also the order of, for example, the reactants here. So they would also have, you can switch orders, and this would also be an augmentation technique.
Gotcha. Thank you.
Thanks. Yeah.
I was wondering if, like, has this been used in practice, or did you actually try to like decompose a difficult molecule into building blocks that you can work with?
What do you mean? If it's, for example, there are open source retrosynthesis software, for example, AC Finder. I have not had the time to play around with it and actually implement it in there. I think they're still using, if I'm not mistaken, they're still using like a standard neural network, and they restrict the templates. They just take templates where they have sufficient amount of data to actually train this. This is the safe way to do it, because yeah, you have training data for those templates, but it can easily be extended with this approach.
I think my question comes from, in the start you mentioned this is like an essential component of a CASP system. Mm-hmm. And I think my question then comes from, how well does like this top-on accuracy kind of correlate to actually finding the full synthesis tree, right? Because there's multiple, there's more to it than just the top-on accuracy. But yeah, I understand that you didn't get to actually implement that, but that's where.
Yeah, it would be very interesting. So as I mentioned here, there are papers mentioning this. This does not correlate that well. But yeah, the safest way would be, would just do this. For example, there's this task of solved synthesis routes. So here you would want all the methods to give you some sort of synthesis route with I don't know, a library of building blocks that you can buy, right, or that you just take for granted because they're very simple solvents. Yeah, so if you run these tasks, there are also certain caveats with this, because if you have a method that, how do you say this? If you have a method that is not very strict, and this is, for example, just giving you a lot of different pathways, there might be that, like, the chance is very high that it arrives at the pathway where it has these building blocks. Whereas if you have a very restricted algorithm or single-step retrosynthesis, which just gives you paths where it really thinks it's doable, then you might not have a diverse set. And the thing to actually then check this is to actually do the synthesis, which is very costly.
Yeah, makes sense.
The final evaluation, but usually they would look at if they theoretically could solve a given target molecule. Yeah, we haven't unfortunately had time to test this, but it would be very much interesting.